Okay, so good evening, everyone. So, so let's wait for 4 to 5 minutes and then I'll start today's session.
राइट साइड में पूरा बाजार के अंदर। अच्छा, एक बार देखो, चार चार पूछे चला जाए, अगर मिलेगा
Okay, so good evening, everyone. So I hope all of you have gone through the week one lecture of this NPTEL course, Genetic Engineering. Akansha, you want to say something? Okay, so I will start presenting the slides. Okay, so I hope the screen is visible. Okay, hello everyone. I'm Akash Jyoti Pathak. I'm one of the PMRF TAs for this NPTEL course, Genetic Engineering Theory and Application. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. And I am Akash Jyoti Pathak, and I am a PhD scholar in the Department of Biosciences and Bioengineering at IIT Bombay. So, uh, so let's first have a quick recap of what all was covered in week one. So in week one, basics of biotechnology, like you all must have learned about biotechnology in your class 11th and 12th. So basic of what is genetic engineering in genetic engineering, and then then
इसलिए प्रैक्टिकल भेजे हमने ना
and then a little bit about cell structure and function was also discussed, like what is the structure of prokaryotic cell, eukaryotic cell. So in eukaryotic cell, what are the differences between animal cells and plant cells? And then the different cell organelles that are present, like nucleus, mitochondria, cytosol, lysosome, endoplasmic reticulum, Golgi apparatus, all these different organelles were discussed. And other than that, self-fractionation topic was also discussed. So in self-fractionation, there are two methods by which you can isolate the different organelles in a cell or different components.
Centrifugation, gradient, density gradient.
Yeah, yeah, differential centrifugation and density gradient centrifugation. Isn't it? If anyone wants to say something. Okay, so you all know what is genetic engineering. Can someone explain in their own language what is genetic engineering?
Hello, alteration of genetic material of an organism to get product, needed product, exploitable product.
Yes, right answer. Any other explanation? Any other definition? Yeah, so he's correct that any organism, if we alter the genetic makeup of any organism, so that it can benefit to our benefit. So alteration in the genetic makeup of any organism to our benefit or for the synthesis of any product or synthesis of any drug, then that is called genetic engineering. Okay, so the different types of bi we can use different host cells, right? We can use bacterial cells, we can use um, plant cells, and then yeast cells. So there are different host cells whose genetic or makeup which can be altered, and we can clone our gene of interest in the gene or in the plasmid, or for bacteria we can clone it in the plasmid of bacteria, and then depending on that, we can see our desired product. Okay. So you all must have all must be knowing about insulin, right? So insulin is a peptide based. It is a peptide based drug, and this insulin is a hormone, and this insulin is now produced by cloning the gene of this insulin uh, in the bacterial DNA, right? So okay, so let's start with the basic topics. So what are prokaryotic cells? So you all know about all these topics. So from class 9, 10, 10, we have been reading about all these. So prokaryotic cells means pro means primitive and karyon means nucleus. So primitive type of nucleus. So the organisms which are having primitive type of nuclei are called prokaryotic organisms. And so if you see a prokaryotic cell, so the best example of organisms having a prokaryotic cell are the bacteria, right? So they are the bacteria, and here is a figure of the bacteria. So you can see that as bacteria will have a cell wall, and then inner to the cell wall it has a plasma membrane, and then inner to the plasma membrane, there is the cytosol, and in the cytosol, different other like ribosomes. There are no membrane-bound organelles in prokaryotes, but certain organelles like ribosomes and nucleoid. We do not call it nucleus because nucleus must have a nuclear membrane, double-layered nuclear membrane, um, it must be surrounded by a double-layered nuclear membrane, and it must have a uh, it has a nuclear envelope. All these structures are lacking in bacterial cells. So the structure of the nucleus so is called nucleus. So nucleus means nucleus-like. So it is not exactly the nucleus, and so these are the different structures which are present in a bacterial cell. So the DNA is what it is smaller. It has around 0.6 million to 5 million base pair, and it is usually single. It has a single chromosome. It is single. It is circular DNA, and it is the size of a prokaryotic cell is much smaller compared to a eukaryotic cell. So other than these, outside the cell wall, there can be a capsule. So capsule has different other properties, confer different properties. So not all bacteria have capsules. There are certain bacteria which have capsules, which confer the bacteria pathogenicity, or it can help the bacteria to attach to different substrates. So these are some of the structures which are present in a prokaryotic cell. Now answer this question. So the form of bacterial genome. So what is the form of bacterial genome? Is it a circular RNA, or is it having multiple chromosomes, a single chromosome, or plasmid only? So just now we discussed that the bacterial genome is what? So who can answer this? Which will be the correct option for this question?
A circular
Sorry. Sorry, can you say any one of you speak at a time? So it will be what?
single chromosome, a single chromosome.
Yeah, it will be a single chromosome. Okay, it will be a single chromosome. So bacteria have the genetic material of bacteria is what? It is DNA. So option A is wrong, right? And bacteria have they do not have multiple chromosomes. So unlike humans or eukaryotes, which have multiple chromosomes, the bacterial genome has a single chromosome. And other than the genome of the bacteria, they have some extra chromosomal DNA, which are called the plasmids. So plasmids only is also wrong because they can have a plasmid, or they might not have a plasmid, but they will never have only plasmids. Along with the plasmid, they will have their own chromosomal DNA. Okay. Okay, so these are easy questions. So the first lecture only basics of biology, like whatever we have read in class 11, 12, or 9, 10 that were covered. So these topics do not require much understanding of the concepts. Okay, so we will quickly go through them. Okay, the answer is option C. Now, the process that allows the bacteria to move towards glucose molecule is what? So option is thermotaxis,
Chemotaxis.
phototaxis. Yeah, answer is what? Chemotaxis. Isn't it? So, so bacteria have a structure called flagella, right? So this flagella helps the bacteria to move from one location to the another, helps in locomotion of the bacteria. So, and glucose is the what? Glucose is a chemical substance, right? It is a chemical substance. So this chemical substance is causing the bacteria or the the bacteria to move from a location of a location where there is less molecules of less concentration of a particular chemical to a more concentration of a particular chemical. So that is called chemotaxis. So when the movement is occurring or the locomotion is taking place on the basis of the chemical concentration of a particular or the concentration of a particular chemical substance, then that is called as chemotaxis. So yeah, so chemotaxis is the process by which bacteria sense and move in response to chemical signals in the environment. So the bacteria has this ability to sense different chemicals which is present in its environment, and this enables the bacteria to move from one location to another. So this is called, when the locomotion is taking place on this basis, it is called chemotaxis. And so when a chemical like glucose is present, the bacteria can detect its concentration using specialized transmembrane receptors. So they have specialized receptors which are called the chemoreceptors, and these receptors allow the bacteria to detect the chemical substance. So it helps that bacteria to detect the glucose and adjust the rotation of its flagella to move towards higher concentration of glucose following the gradient. Okay, so these are easy concepts. So let's move. Now, eukaryotic cell, there are mainly in eukaryotic cell, we mainly learn about two types of eukaryotic cell. One is this animal cell and another is the plant cell. Okay, so this figure has been taken from this textbook, Cooper Cell Biology textbook. You all must be have reading this classical textbooks in your BSc and MScs. So uh, so you can see here that bacteria that this is the animal cell, right? This is the figure of an animal cell, and on the right is the figure of a plant cell. So you can see that animal cell, the most striking difference is the cell wall. The cell wall is present in bacterial in plant cell, while it is absent in animal cell. And also, what are the other differences that you can spot in these two figures of animal cell and plant cell? That this vacuole in a plant cell is very large, and the plant cell have chloroplast, which allows the plant cell to perform photosynthesis. These are lacking. And then there are the centrioles in animal cells. Okay. Plant cells also they do not have the centrioles, right? There's only the animal cells will have centriole, isn't it? Then there are this endomembrane structure, like the endoplasmic reticulum, vacuole, Golgi bodies, all these structures they together form this endomembrane system. And then there are cytoskeleton, which provides shape and structure to the animal cell. So all these different organelles are present: endoplasmic reticulum, Golgi bodies, then mitochondria, peroxisome. So now, what are the differences between prokaryotic cell and eukaryotic cell? So the first difference is that the prokaryotic cells are small. They are smaller. So their size is around, with their size is around what? around 1 to 2 micron of a if we consider a bacterial cell, then its size is around 1 micrometer. And eukaryotic cell have around 10 to 100 times larger size. They are of having a diameter of around 10 to 100 micrometer. And the cytoplasmic organelles are absent in prokaryotic cell while they are present in eukaryotic cell. The DNA content is so 1 million to 5 million base pairs in prokaryote, while in eukaryotes they're in a few millions, 6.6 into 10 to the power 9 million into 10 to the power 9 base pairs in humans. No, and then the chromosomes as just now we discussed that the bacterial cell have a single chromosome while multiple chromosomes are present in eukaryotes. So what are the different? So you can read all these text. So there are these are from the classical textbooks that we follow in our bachelor's and masters. So what are the features of eukaryotic cell that are not present in prokaryotes? So there is division of cell into nucleus and cytoplasm separated by a nuclear envelope containing complex pore structures. So there is a specific new compartmentalization that we see in eukaryotic cells. They have a nucleus, they have a cytoplasm, and which is lacking in prokaryotic cell. Okay. Then there are complex chromosomes. So the DNA, if you look at the DNA of eukaryotic cells, then the DNA remains wrapped around this protein complexes to form structures called nucleosomes. And while prokaryotes have naked DNA, which means that only the genetic material is present, and this is not wrapped around. So all these things you know, right? The DNA is not associated with any protein, isn't it? So there are complex membranous cytoplasmic organelles that are present in eukaryotic cells. These are endoplasmic reticulum, Golgi complex, lysosome, endosome, peroxisome. And then there are specialized cytoplasmic organelles for aerobic respiration, mitochondria, and in plant cells there are chloroplasts. Okay. Okay. You can read all this. I'll share these slides and you can read all these. So eukaryotic cells will have they will have cilia, complex cilia, flagella, right? So if you see different eukaryotic cells, like the sperm cell have flagella, and then there are cilia um, in our intestine. There is cilia in our gut. Also, there are cilia-like structures. So there are these are complex. If we look at the structure of the flagella and cilia in a prokaryotic cell and eukaryotic cell, these also differ. These structures differ, and presence of 2 copies of gene per cell. Then sexual reproduction is seen while in bacteria, we all know that the binary fission and other different types of asexual reproduction takes place. Okay. Okay. So you can go through this. You all already know all these topics. So now answer this question: the structural component that is shared by both plant and bacterial cells, but not animal cells. So what is that structural component? So these are easy questions, right? Class 9, 10 level question. So what is that particular component? So nucleus, chloroplast, cell wall, lysosome. What
Cell
is the
wall.
answer? Cell wall. Right. So
Cell wall.
cell wall. So the plant and bacterial cells both will have cell wall, but the animal cells do not have cell wall. Okay. Okay, so all these questions do not require any explanation. You already know about this. Now, bacterial cell wall, you all know about bacterial cell wall. So the bacterial cell wall, if we look into the details of what is present in a bacterial cell wall, so there are 2 types of bacteria, right? Gram positive and gram negative. So you must have all performed the experiment of gram staining. And what is the basis of calling a bacteria gram positive or gram negative? Is that the gram staining method. So if a bacteria takes up or is gives positive result in gram staining experiment, then we call it as gram positive. It gives negative result, or the crystal violet or the blue color dye that gets washed off, then we call it gram negative. And what is the basis of why a particular bacteria takes up the stain or retains this crystal violet stain, while the other group of bacteria, gram-negative bacteria, do not retain this stain, is because of the structure of their bacterial cell wall. So the bacterial cell wall is made up of this chemical substance called peptidoglycan. Okay, we all know about this, isn't it? Peptidoglycan layer, and this peptidoglycan is it is what? It is a heteropolysaccharide. Okay, so you must have read about this structure in your biochemistry classes. So it is a heteropolysaccharide. Why? Because it is made up of this two basic subunits, NAM and NAG, N-acetyl muramic acid and N-acetyl glucosamine. So the repeating units of NAM and NAG gives rise to this structure, which is called the peptidoglycan. Basically, the bacterial cell wall lies outside the plasma membrane. So you have the plasma membrane, and then outside the or outer to the plasma membrane is the bacterial cell wall. Right. They have the bacterial cell wall, and this bacterial cell wall is made up of a particular substance which is called peptidoglycan. So the chemically, this peptidoglycan is made up of the NAM and NAG subunits. And what is the chemical bond which is present between the NAM and NAG residues? It is beta-1,4 glycosidic bond. Okay, so here you can see the structures. This is this yellow colored structure. This is N-acetyl muramic acid and N-acetyl glucosamine. And then there is a peptide chain which is seen attached to the N-acetyl muramic acid. So there is L-alanine, L-amino acid, D-amino acid. Then again there is pymalic acid, then D-alanine. So the one thing to note over here is that the bacterial cell wall have D type of amino acids, right? But human cells or eukaryotic cells do not have. We all have what? We only have L type of amino acids. So the D type of amino acids is only seen in bacterial cell wall. And so you remember this, what type of glycosidic bond? So this bond that you see, this is called the beta-1,4 glycosidic bond. And the NAM monomer, these are a few details about this peptidoglycan structure. So this NAM monomer is attached to tetrapeptide side chain. So this tetrapeptide, because you can see there are 4 different amino acids, this L-alanine, D-glutamic acid, pymalic acid, and D-alanine. Okay. And then also what happens is D-alanine may be attached, may be connected to the peptide interbridge or to the diaminopymalic acid in another tetrapeptide chain. So you can see here this gray colored rod-like structures which is connecting one NAM unit to another NAM unit which is present in another strand. So by this inter-strand cross-link, what it is doing is it is creating a mesh-like structure and providing structural support or integrity and structural support to the cell wall, basically. Okay, so you remember this, what type of glycosidic bond is present and what are the subunits that is composed of bacterial cell wall, NAM and NAG. So now if you look at the structure of the cell wall, so the peptidoglycan layer that is present in gram-positive bacteria and a gram-negative bacteria, then you can, looking at this figure, you can see that the peptidoglycan layer is much thicker in gram-positive bacteria. So this layer is the peptidoglycan layer. It is much thicker in gram-positive bacteria compared to that which is present in gram-negative bacteria, which is much thinner. And another striking difference between the two types of bacteria is that outer to this peptidoglycan layer, there is an outer membrane. There is an outer membrane, while this outer membrane is lacking in gram-positive bacteria. So if you look at a gram-negative bacteria, then you have an outer membrane. Then inner to that, you have a peptidoglycan layer. And then there is an inner membrane. Okay, then there is an inner membrane, and then inner to the inner membrane, you have a cytosol. So the space between this outer membrane and peptidoglycan layer, or the peptidoglycan layer and inner membrane, these are called the periplasmic spaces. Okay. So all these have been, you can see in this figure. So this periplasmic space is the space between this outer membrane and peptidoglycan layer and the inner membrane and cytosol. So if you look at the, there is another difference in the, that is the difference in the periplasmic space of a gram-positive and gram-negative bacteria. So the gram-positive bacteria, you can see because of this thick peptidoglycan layer, the periplasmic space is much reduced. While because of the thin peptidoglycan layer in gram-negative bacteria, the periplasmic space is higher, is larger, or it is wider compared to gram-positive bacteria. And also these gram-negative bacteria, they have different types of membrane proteins in the outer membrane. So porine, lipopolysaccharides. So all these different structures which are present in the outer membrane confer pathogenicity to the bacteria. So these are the foreign substances which are recognized by the immune cells, or they help the immune the human cell, the immune cells to detect them as foreign and confer immunogenic response. Okay. So yeah, this is from this book, My Prescott, which you must have studied in microbiology classical textbook. So you all know about this gram positive and gram negative. That gram positive stains purple and on staining with this crystal violet, and they have thick cell wall, which is composed primarily of peptidoglycan. So here, this is an electron micrograph. You can see here that the peptidoglycan layer is much thicker, while this gram negative have this thin peptidoglycan layer. So this is a schematic. So they have thin peptidoglycan layer. Then there is an outer membrane, and then there is a plasma membrane. So this periplasmic space in gram negative is also very. So you can look at this figure, and you can understand what are the difference in their structure. Okay. The outer membrane also has different structures, as I was mentioning, lipopolysaccharide. And one more substance that is present is the teichoic acid. Teichoic acid is present in gram-positive bacteria, while it is absent in gram-negative. Okay, so now answer this question. So just now we discussed the linkage that connects the N-acetyl glucosamine and N-acetyl muramic acid.
Beta 1,4 linkage.
Beta 1,4 linkage.
Beta 1,4 linkage. Okay, the answer is option B. Okay, so this is directly from the lecture slides. So you can also see here that this N-acetyl and N-acetyl glucosamine and N-acetyl muramic acid, which is linked by beta 1,4 linkage. Okay. We already discussed this figure. So let's move on now. Now, coming to mitochondria, we all know about mitochondria, right? So they are double membrane structures, and they are the powerhouses of the cell. And so because they're double membrane, they have an outer membrane, they have inner membrane. The inner membrane is folded, right? And these fold-like structures, these are called the cristae. And also this mitochondria plays an important role in metabolism. Okay. So in glucose metabolism and in glycolysis, you must have read about glycolysis pathway. So when this glucose is broken down into pyruvate, and then the pyruvate moves from the cytosol into the mitochondrial matrix. So it moves to the mitochondrial matrix, and then the pyruvate is converted into acetyl-CoA. This acetyl-CoA moves into the citric acid cycle. So all the different enzymes that are present, that are required during glycolysis, or this TCA cycle, not like the TCA cycle or the citric acid cycle, they are all present in the mitochondrial matrix. So this different enzymes break down this acetyl-CoA into this carbon dioxide, and then the other reducing agents, that is NADH and FADH2. These are also produced, and this NADH and FADH2 then they can be again oxidized, and through this electron transport chain, which is present in the inner membrane of the mitochondria. So this electron transport chain consists of the several protein complexes, complex 1, complex 3, 4, 5, all these different agents and protein complexes. And then there is this ATP synthase, which breaks down this gradient. So all these you will be reading in biochemistry. So these details are not required for this course. So just basic is that it has this outer membrane, inner membrane. Inner membrane is folded into crista and structures which is called cristae. And then another thing is that the oxidative phosphorylation or this electron, the proteins which are involved in this electron transport system, they are present in the inner membrane. They're present in the inner membrane, but so the outer membrane is more permeable. So it has certain protein complexes which makes it permeable, while the inner membrane is not less permeable, and there are these protein complexes are present, and this matrix is the has several enzymes which is involved in metabolism. Okay. So these are the few of the points which you need to remember. So outer membrane are permeable to small molecules, and this outer membrane contains channels. So there are channel proteins which are present in outer membrane. These channel proteins are called porins, and these porins allow free diffusion of molecules which are less than 100 Dalton. And this inner membrane forms cristae which extend into the interior. So what is the purpose of this cristae? Why is it folded in such a manner? Anyone? What would have happened if it was if it was just a circle or a disc shape? There were no invaginations or infolding of the inner membrane to form such structures. If it would be flat, then what would have
area
happened?
for reaction.
Yes.
Surface area for reaction.
Yes, correct answer. It increases the surface area for so once it is folded, then in these folds it increases the surface area so that there can be more number of protein complexes, and the electron transport chain or system or electron transport chain reaction that takes place, that can occur more efficiently. So yeah, you answered correctly that this reaction that is taking place, that will become more efficient. Okay. The matrix of the mitochondria, it contains several enzymes for this TCA cycle and oxidative breakdown of fatty acids. So oxidative phosphorylation takes place in inner mitochondrial membrane. And another thing is that this mitochondria is always inherited from mother. There's human maternal lineage, and it is thought to it is endosymbiotic origin. All these things you must have read about. So it is thought that it has evolved from bacteria because it has its own genetic material. It has its own ribosome, right? 55S ribosome is also present. So all these is a semi-autonomous organelle. It can perform its own replication. It can perform its own um, all these it can perform its own reaction or chemical reactions that can allow it to survive. So it can also generate its own energy. So that is why it's called as a semi-autonomous organelle. And so it has so according to this endosymbiotic theory, it is proposed, or it is believed that it must have at some point in of time in evolution, it must have existed as a free structure, um, or in the environment, but eventually it got engulfed by a eukaryotic cell, and then they started sharing a symbiotic relationship with each other. So that is what it is. Okay. So what are the contribution of mitochondria to the immune responses? So this question, this was discussed in the lecture. So how does the mitochondria contribute to the immune response? So by producing antibodies, by storing white blood cells, by generating reactive oxygen species in immune cells to kill infectious agents and produce.
C.
Yes, answer is option C. Yes, correct. So can you explain this? Why? How is it generating reactive oxygen species?
It actually produces
Yes, because this electron transport chain as reaction is taking place in the mitochondrial inner membrane. So this oxygen, which is released, that actually gets. So ultimately, this electron is accepted by water and it gets converted into oxygen, right? So what happens is basically the mitochondria are the major site of cellular reactive oxygen species production. And how is this ROS produced? So this ROS is produced due to this electron transport chain, which is present in the inner mitochondrial membrane. And this inner mitochondrial membrane drives the generation of ATP. So the electrons are passed through several protein complexes, and sometimes the electron leak prematurely and react with oxygen, primarily at complex 1 and complex 3. So when this electron reacts with oxygen, it results in the formation of the superoxide anion, O2 minus, superoxide anion, and this is the primary mitochondrial reactive oxygen species. And this reactive oxygen species is used by the immune system as a weapon against microbes. Okay. So this is again from this lecture slide. So what are the different functions of mitochondria? It helps in production of ATP. So I'm not going into the details of this electron transport system and oxidative phosphorylation. What are the different complexes? So that is not part of this course, the part of biochemistry. So all these just you can understand that because of this presence of this electron transport chain which is taking place in the inner mitochondrial membrane, there is this oxygen that is released, and also the transfer of electron from one complex to the other complex. This electron reacts with this oxygen and results in the generation of a reactive oxygen species. Okay. And how this reactive oxygen species, reactive nitrogen species, how they are formed, how these neutrophil, macrophages, they have all these different species. So those are, if you go into detail, then there are a lot of concepts in this also. This how the immune cells are have this specific ability to generate this reactive oxygen species. Okay. Okay. So now let's discuss this one particular topic, apoptosis, because there are a few questions related to apoptosis. So you all know what is apoptosis, right? So apoptosis is programmed cell death, and so basically it is one of the uh, survival or this is a very important process in development as well. So during development, the when our appendages are being formed, if there are certain cells which or the separation that is present between our fingers, that separation is necessary, and for that separation there must be particular the cells in these regions must be destroyed. So they must undergo cell death. So that is called apoptosis. So why is it called programmed cell death? Because um, this type of death does not occur spontaneously. There is a particular sequence of events that take place for during the cell death. So that is why it is called programmed cell death, because there is a series of cellular changes that take place. So what happens is, you can see, though, this is a normal cell which is having this plasma membrane, and inner to this, the nucleus, which is having the genetic material, the DNA. And so in the first step, what happens? The DNA gets fragmented. DNA gets fragmented means what? The DNA gets broken up. So there are different enzymes which causes uh, the DNA to get chopped into different fragments. And then after DNA fragmentation, what happens? Chromatin condensation. So the different DNA fragments, they get condensed. And then in the next step, what happens? There is fragmentation of nucleus. So you see here that the single nucleus is now fragmented into 3 different nuclei. And in the next step, what happens? There is fragmentation of the cell, and the plasma membrane also has, you can see that there is blebbing of the plasma membrane. So it is not uniform. It has developed some irregular structure, and gradually the cell gets phagocytose. Okay, finally, what happens? The cell itself shrinks and breaks up into membrane enclosed fragments called apoptotic bodies. Okay. So you see here, so why is it so? This is called apoptosis because you can see that there are what is happening is a series of chemical events or chemical changes or morphological changes are taking place one by one. So first, what will happen in the first step, there will be DNA fragmentation, chromatin condensation. Then in the next step, there will be fragmentation of nucleus. And then in the third step, fragmentation of cell. And then the cell will shrink and break up into membrane enclosed fragments. So one by one, each of the steps are taking place. Okay. So that is why it is called programmed cell death. Now, one of the important layer that is present is the caspases. So there are a lot of details about this caspases in any cell biology book like Albert or Hooper. You can get the details of these caspases. But for this lecture, a little understanding of caspases is required because a few topics were covered in week 1. So I'll not go into too much detail. So caspases are the main players of the apoptosis process. So what do caspases do? You can see here they inhibit the caspases cause they are inhibitors of DNase. Or then it causes fragmentation of nucleus. So these caspases acts on nuclear lamines. So nuclear lamines are the cytoskeleton filaments which are found inert to the nuclear envelope. So these caspases target the nuclear lamine and cause their destructions. And because the nuclear lamines gets fragmented, the nucleus also gets fragmented. Then the caspases target the cytoskeleton proteins. So these are the different targets of caspases which have been shown in this figure. So it targets the inhibitor of DNase. So once it targets the inhibitor of DNase, what will happen? The DNase will get activated, right? So if you target the inhibitor of DNase, the DNase will get activated and the DNase will now start to degrade the DNA. It will start to fragment the DNA. Then it can target the Golgi matrix proteins and then scramblase. Scramblase is so we will come to this topic in plasma membrane. So it has different types of these phospholipids. So if you have a little bit knowledge about cell biology in your bachelor's, then you must be knowing about this role of phosphatidylserine. So it is present in the inner leaflet, and once this phosphatidylserine gets translocated from the inner leaflet of the lipid bilayer to the outer leaflet, then it acts as a signal for cell death or eat me signal. It acts as a signal which is called eat me. Okay, so what are basically caspases? They are a family of proteases. So they are a family of proteases which bring about the events of apoptosis by digesting the cell proteins. Proteases. So what are proteases? They will digest the proteins. So those are called proteases. DNases are what? They'll digest the DNA. So caspases are basically proteases. And why are they called caspases? Because if you look at the structure of this caspase protein, then they have a cysteine residue at their active site and the target protein that it degrades. This target protein, there is a particular site at which this caspases act. So in the target protein, this caspases specifically cleave after the aspartic acid residue in the substrate protein. That is why they are called caspases. So they have cysteine residues at their active sites and they cleave the aspartic acid residues in the substrate residue after the aspartic residue. Okay, so all these different so they can cleave over 100 different cellular proteins, and by cleaving these 100 different cellular proteins, what it is ultimately causing, it is causing the morphological and alterations which are characteristic of apoptosis. So if you remember the figure that I showed just now, so you can see that how the morphology is changing. Morphology of the cell is changing in response to apoptosis. So all these morphological changes are brought by this caspases. Okay, so they are degrading the proteins that are present in nucleus, that are present in cytoskeletal proteins, that are present in Golgi matrix proteins, and as a result, and it is resulting in the cellular death. Okay, so in addition, caspases cleave nuclear lamines, leading to fragmentation of the nucleus, the cytoskeletal element proteins. So this figure that I was explaining, the cytoskeletal proteins, which leads to disruption of the cytoskeleton, blebbing of the plasma membrane, cell fragmentation, the Golgi matrix proteins, leading to fragmentation of the Golgi apparatus, and finally, what it causes is that the cell gets shrunk and it breaks up into membrane enclosed fragments called apoptotic bodies. Okay. So now there is one more question which is related. That is why I'm discussing this topic. There are different pathways of apoptosis, which you will be reading in detail in cell biology courses. So there is this intrinsic pathway and extrinsic pathway. So in this lecture, I'll just give a brief idea about the mitochondrial pathway of apoptosis, how the mitochondria is involved in apoptosis. So there are actually uh, two types of factors which control. So whether a cell will go into apoptosis or whether it will not go into apoptosis is determined by a couple of factors which are present in the cell. So there are two types of factors which are called the pro-apoptotic factors, and another is the anti-apoptotic factor. Okay. So in apoptosis, what has to be done is the ultimate goal is to activate a caspase. So these caspases need to be activated. Basically, the caspases remain in procaspase form. Okay. So the main goal of apoptotic pathway is to activate the caspase. So there are different type of caspase, caspase 3, caspase 9, and there are multiple different types of caspases based on which type of protein they are acting. Okay. Now, so these pro-apoptotic and anti-apoptotic factors, these control so a sequence of events takes place before this caspase gets activated. Okay, so in this figure you can see that there is a cell death signal. Okay, so once there is a cell death signal, it activates the pro-apoptotic effector proteins. So the pro-apoptotic effector proteins are back and backs. So these back and backs. So this figure is taken from this book, cell biology book, Cooper. These are pro-apoptotic factors, and they come under the BCL2 family of proteins. So these backs and back, you can see in this figure, these proteins are mainly present in the outer membrane of the bacteria. Okay, now what happens once there is a cell death signal is received by this mitochondria, then what happens is this backs and back protein, they starts to oligomerize. So you can see here that they have formed an oligomer structure. Now, once this oligomeric structure is formed, what happens is there is a particular molecule which is present in the intermembrane space. Intermembrane space is the space between the outer membrane and the inner membrane of mitochondria. So this molecule which is present in the intermembrane space is called the cytochrome. So once this backs and back forms an oligomer in the bacterial outer membrane, these cytochrome molecules are released from the intermembrane space into the cytosol. Now, once this cytochrome C is released into the cytosol, it goes and binds to this APAF1 complex. So this is the protein complex, and you can see here in this protein complex, which is called APAF1, the cytochrome C molecules have bound to this APAF1 complex, and this structure is called apoptosome. Now, once this apoptosome is formed, what it causes is it activates this apoptosome, then converts this procaspase 3 into this active caspase 3, and once this this caspase 3 is activated, then all these different proteolytic degradation starts to take place. Okay, so let's just look at the steps one by one. So in mammalian cells, members of the BCL2 family act at mitochondria, which play a central role in controlling programmed cell death. So when activated, what happens? The pro-apoptotic effector proteins, backs and back, oligomerize to form pores in the mitochondrial outer membrane. So once this that signal is received, the back and backs, they form an oligomeric complex on the mitochondrial outer membrane, and this leads to the release of cytochrome C from the mitochondrial intermembrane space. Then once this cytochrome C is released into the cytosol, what happens? The key initiator caspase in mammalian cells. So there is initiator caspase and then there are effector caspase. So there are a lot of theory that is related to caspase. What are the pro-apoptosis, that initiator caspase? What are the effector caspase? So these details you will be reading in your cell biology course. But what happens here is that there is an initiator caspase which is called the caspase 9, and this caspase 9 is activated by forming a complex with APAF1. So this is the APAF1, and you see here this is the caspase 9. Together they form a complex which is called the apoptosome. Now, what happens is under normal conditions of cell survival. So, in the normal conditions, the cytochrome C remains inside the mitochondrial intermembrane space. Okay, so it remains in the mitochondrial intermembrane space, and this APAF1 and caspase 9, they are present in the cytosol. So this caspase 9 remains inactive. So when the cytochrome C is present in the intermembrane space, then the cytochrome C will not form a complex with this caspase 9 to form this apoptosome complex. So the cell death signal, there will be no initiation of this downstream signaling will not take place. But once this backs and back is activated, then there is release of cytochrome C into the cytosol, and then this cytochrome C goes and binds to this caspase 9 APAF1 complex, and then once this caspase 9 uh, uh, is activated, this caspase 9 will again, what will it do? This is the initiator caspase. It will go and cleave and activate downstream effector caspases such as caspase 3. Caspase 3 eventually resulting in cell death. So sometimes there are questions that comes in objective questions like what is the initiator caspase which is present in the mitochondrial pathway of apoptosis. So the initiator caspase is caspase 9, and what is the effector caspase? So effector caspase, what do we mean that what is the ultimate or the main caspase which is involved in causing apoptosis of the cell? So that is caspase 3. Okay. So now answer this question. So what is the role of mitochondria in apoptosis? So role of mitochondria in apoptosis. What? Which option? Yeah, which option?
D option.
Yeah, option D. So just now we discussed everything about these caspases and all the different signaling molecules which are involved. Okay. So in apoptosis, what are the main players? They are caspases, right? So this much we can all remember that the main players of apoptosis are caspases. So the only option that is here is the option number D. So the answer is option number D in response to cell surface signaling. Okay. So how this signaling happens, there is a lot of details that you can go through from any standard book. Okay, so the answer is option D. So apoptosis, so this is from the lecture slide. So in an adverse event of exposure of cell to cytotoxic agent or environmental condition, what happens? The cell surface signaling gets activated, and once this cell surface signaling and activates cytosolic caspases, and ultimately these cellular events activates DNA's activity within nucleus and degrades genomic DNA to cause cell death. Okay. Okay. So these points are clear. Now answer the next question. So the enzyme activity that is ultimately activated in the nucleus during apoptosis causing cell death. So which is the enzyme or sorry? DNase, right? DNase. DNase. Answer is DNase. So just I showed you the figure in the initial slide that what happens ultimately the DNA gets fragmented. And also in this slide also I showed in this slide also that these caspases act on the inhibitor of DNase. So once it acts on the inhibitor of DNase, then the DNase will get activated because there is no protein or no inhibitor to inhibit the activity of DNase. So the DNase gets activated and it will lead to fragmentation of the DNA. Okay. Answer is DNase. Okay. Now, this is a very easy question actually. This is very logical question that why animal cells contain more mitochondria compared to plant cell. All of you know this. The options are animal cells are more energetically active. Animal cells have a cell wall. Animal cells perform photosynthesis. Animal cells are larger.
A.
Yeah, so option
A.
A. Okay, so this is so yeah, obviously that why would a cell need more mitochondria? Because if a cell has to is requiring more energy, then it will have more mitochondria. Like muscle cells have more mitochondria. So similarly, animal cells have more mitochondria because they are energetically active. Okay. Okay, now these things also were discussed. So what are endoplasmic reticulum? So endoplasmic reticulum is one of the largest organelle. If we look into that, the area through which it is spread throughout the cell, then the endoplasmic reticulum is actually, if you look at the structure of cell, this membranous structures remain is widespread throughout the cell. So that is why endoplasmic reticulum basically forms a network of membrane enclosed tubules and sacs that extends from the nuclear membrane throughout the cytoplasm. So starting from this nucleus and towards this and almost towards the periphery, it forms a network of network-like structure, and the entire endoplasmic reticulum is enclosed by a continuous membrane. It is enclosed by a continuous membrane, and it is the largest organelle of most eukaryotic cell because it is spread throughout the cell structure, internal structure of the cell. So there are two types of endoplasmic reticulum: this rough endoplasmic reticulum, smooth endoplasmic reticulum. This rough endoplasmic reticulum means what? That this endoplasmic reticulum will have ribosomes on its surface, and the smooth endoplasmic reticulum are the ones which do not have ribosomes on its surface. And what is the function of RER and SER? All these things you know, right? We have been reading since class 9, from class 9 to and throughout we have been reading all these concepts. So they are involved in protein synthesis, and this smooth endoplasmic reticulum involved in lipid synthesis, and heparin, sterols, and other molecules get synthesized in this smooth endoplasmic reticulum. So it is involved in lipid rather than protein metabolism. And also this ER is the site of protein folding. Another important point is it is the site of protein folding, assembly of multi-subunit protein, disulfide bond formation, end-link glycosylation, addition of glycolipid anchors to some plasma membrane protein. So all these things. So why is it a site of protein folding? Because this basically this endoplasmic reticulum provides the oxidizing environment. So why is an oxidizing environment necessary for protein folding? Because you must have read about this particular amino acid cysteine, right? Cysteine forms these disulfide bridges or disulfide bonds between. Uh, so one cysteine molecule can form a disulfide bond with another cysteine molecule, and this disulfide bonds helps in the folding of the protein, or it gives a proper conformation to the protein structure. So that this oxidizing environment is necessary for the formation of these disulfide bonds. Okay, because the structure is as CH2SH, and in the oxidizing environment, this H gets replaced and it can form bond with another CH2 group. And here you have this C alpha backbone, whatever. Okay, so basically that provides this oxidizing environment. As a result, it is a site of protein folding and disulfide bond formation as well. There are other enzymes also which are involved in disulfide bond formation. So what is the role of endoplasmic reticulum in calcium homeostasis?
Calcium +2 sequestration.
So calcium sequestration. So basically, endoplasmic reticulum is a storehouse of calcium. So it stores calcium inside the calcium is stored inside the endoplasmic reticulum. So that is why it is called calcium sequestration. Okay, and this calcium is involved in several signaling mechanisms which you will read about in the cell signaling. You must have read that what happens when this calcium is released, and then other downstream signaling pathways that take place. Okay, GPCR and all these you have must will be reading about them. So it is basically it is a major repository of intracellular calcium, and it also plays a key role in signal transduction. So in cell signaling, you will read about how this calcium plays a role in cell in signal transduction. So what are the different? So this is from the lecture slides. You can read about what are the different functions of this endoplasmic reticulum. So basically, this is the function of smooth endoplasmic reticulum is that it is involved in synthesis of steroid hormones in gonad cells, detoxification because it has this P450 cytochrome oxidase. This enzyme is present that can cause detoxification, and then calcium sequestration, and then synthesis of protein, phospholipid, carbohydrate, protein sorting to different organelles. Right, so protein sorting and all these you will read in cell biology, so how this protein sorting takes place. Then protein modification such as glycosylation also takes place in endoplasmic reticulum. Okay, now coming to lysosome. Okay, so what are lysosomes now? So lysosomes, as you all know about this, they are known as the suicidal bags. Okay, we have been hearing this since class 9 or 10. And so they are single lipid bilayer. And why are they called suicidal bags? Because the lysosomes basically what it does, it stores several hydrolytic enzymes in its lumen. It has so almost all different hydrolytic enzymes are present inside the lysosome, starting from this um, uh, like all the different glycosidases, phosphatases, nucleases, proteases, lipases, all these different hydrolytic enzymes are present inside the lysosomes. And this, as a result, it is involved in macromolecular degradation. Okay. Another important point is that for the uh, this VATPases, there are proton pumps. Okay, the proton pumps, what it does is the proton pumps are present on the membrane of lysosomes, which maintains an acidic lumen inside the lysosome. So why is it necessary to maintain an acidic pH inside the lysosome? Why are these VATPases required? So have you read about these in your cell biology course? That what is the function of VATPases and why is an acidic environment required inside the lysosome? So it is required for the activity of these enzymes. All enzymes have a particular pH and a particular temperature at which they are active. So for the activity of these enzymes, an acidic pH is required. That is why the inside of the lysosome has an acidic pH. Okay. So they are called the suicidal bags because they contain potent degradative enzymes. So all these different enzymes that I mentioned, they're all present in the lysosomes. And this lysosomal membrane permeabilization can release hydrolases into the cytosol. So once this lysosome membrane gets permeabilized, then these hydrolases are released into the cytosol, which triggers the programmed cell death. Why are they called suicidal bags? Because they have the capacity to self-digest the cell under stress. Now, pathogen degradation. So there are structures. So the lysosome, when it engulfs a phagocytic cell or a phagosome. So phagosome is basically a or phagocyte carrying a foreign substance. So that is called a phagosome and lysosome. They merge with each other and they form a structure called phagolysosome. And once this phagosome is engulfed by the lysosome, then the different hydrolytic enzymes that are present allows the bacteria, viruses, and parasites to be digested and destroyed. Okay. And macromolecule turnover. Another function is macromolecule turnover because it helps in degradation and recycling of proteins, lipids, carbohydrates, and nucleic acids. So this is another important property of lysosome is that it replenishes the cell, or it what happens that after a particular time the organelles also they become to age, or they lose their function. So the old organelles need to be replaced by new organelles. The function of clearing of the old organelles uh, is done by lysosomes. So lysosomes also what they does is they can digest the old organelles or the other different organelles which are present in the cell. Okay, and the organelles, any organelle, they are made up of these different proteins, lipids, carbohydrates. So all these different components. So once these different proteases, nucleases will act on the protein and lipid, they will convert them into amino acids, DNA, lipid molecules. So these can be recycled again. Okay. So another important function is the autophagy. So what is autophagy? So it is the sequestration of cytoplasmic cargo into autophagosomes that fuse with lysosomes. So we will discuss about this autophagy in detail in the next slide. Okay. So these points are clear, right? So the lysosomes are popularly known as suicidal bags. Why? Option is because they kill pathogens. Option B, because they produce energy. So you can directly omit this option because the organelle which produces energy is mitochondria, right? Due to their role in autophagy and due to their role in protein synthesis. So basically, they are called suicidal bags because of their role in autophagy. Okay, because autophagy means what? It can the lysosome have the property to destroy its own or destroy the organelles of its own cell. So that is why it is autophagy. Okay, that is why they are called suicidal bags. So this was explained in the lecture also. So they are called as suicidal bags because of their role in autophagy. And what is autophagy? It is a cellular process that operates in cells during starvation to meet their energy requirements. So as I mentioned, lysosomal lumen is extremely acidic and it contains proteases, cytosolic enzymes that degrade the ingested material. Okay, so let's learn about this autophagy. Okay, so this is again from this book Cooper. So what is basically autophagy? So you can see that this is a cell in this figure. This is a cell. This is the plasma membrane, and then inside the plasma membrane you have the cytosol, and then this is the lysosome. Okay, this is the lysosome, and this lysosome, what it is doing in brief, what it is doing, uh, it is merging with the autophagosome. So a structure is formed which is called autophagosome. And when this lysosome fuses with this autophagosome, what happens? So you see here a structure called autophagosome is formed. So what is autophagosome? So for example, if your mitochondria has to be degraded, okay, if the mitochondria has to be degraded, then what happens? This mitochondria gets surrounded by a membranous structure. And this structure that is formed is called the autophagosome. Now, this autophagosome fuses with the lysosome. So once this lysosome will fuse with the autophagosome, then different hydrolytic enzymes which are present inside the lysosome, what will it do? it will destroy the protein components that is present in the mitochondria, the lipid components, the nucleic acid components. All these different components will be degraded. Because of the hydrolytic enzymes that is present in the lysosome, and in this way, it can clear off the worn out organelles or the organelles which have aged off. Okay. So the lysosomes are the organelles which are responsible for autophagy, and there is a lot of research also which is taking place in this field of autophagy, how these autophagosomes are formed, what is the signal which promotes the formation of this autophagy. Um, so, uh, so basically, this is a simplified explanation what happens. Okay, so the turnover of the cell's own components, including cytoplasmic organelles and cytosolic protein. So it is degrading the substances, but at the same time, what is doing? It is not throwing it away, it is recycling the components. So the proteins get degraded into amino acids, and then this can be recycled. Similarly, the carbohydrates can get degraded into glucose, and they can be recycled to form a new carbohydrate molecule or new protein structures. Okay, so that is why it is called the turnover of the cell's own components. Okay. So the region of the cytoplasm or internal organelles are enclosed by membranes derived from the endoplasmic reticulum, forming autophagosome. So as you can see in this figure, so this endoplasmic reticulum forms a forms a membranous structure, and this membranous structure engulfs the organelle which has to be degraded. So for example, the mitochondria has to be degraded, then it forms a membranous structure around the mitochondria, and this structure is called autophagosome. Now this autophagosome, what will happen? It will fuse with the lysosome, and it will form phagolysosome. So once it has fused with the lysosome, this structure is now what it is called? This structure is called phagolysosome. Autophagosome plus lysosome, where it is called as a phagolysosome. And then, in addition, autophagy is activated. Then what will happen? The cellular contents will be digested. Their contents will be digested. Now, another reason of when does autophagy takes place? So when a person is on starvation or in starving cells, because in starvation, the protein and the carbohydrate or the cell is running out of the metabolic energy substrates. So what happens is autophagy can take place, and then the protein and the carbohydrate can be supplied to the cells. Okay, so it also gets activated when the cells are deprived of nutrients. So under conditions of nutrient starvation, autophagy allows cells to degrade the non-essential macromolecules so that their components can be reutilized. So once this mitochondria is degraded, the components that is making of this mitochondria that can be reutilized, isn't it? That is autophagy. So these are the different steps. I discussed the steps. So this is another figure. So you can see here. So this is the endoplasmic. So this is the endoplasmic reticulum, and then membranous structure is formed around this organelle. This is called the autophagosome. This is getting fused with the lysosome, called phagolysosome, and then the contents are getting degraded, and then the contents are released from the cell by exocytosis. Here also the same thing is written, that mitochondria gets surrounded by a double membrane structure and called autophagosome. Then this mammalian cell autophagosome are thought to form de novo from contact sites between ER and mitochondrial outer membrane. Once formed, the outer membrane of the autophagosome fuses with lysosome, generating a structure called autolysosome. It is sometimes called autolysosome, it is called phagolysosome. So there are different names of the same thing. And the enclosure of the protease gets degraded. Okay. So this is already okay. So this is already I have forgot to add the animation. So the cellular process that are supported by lysosome during starvation is what? So it is. So during starvation, the process that is supported by lysosome is what? Just now we discussed, it is autophagy. Okay. So what is autophagy? So yeah, if a population of cell, whether it is derived from yeast, plants or animals, if it is placed under starvation conditions, then what will happen? There will be a marked increase in autophagy. And under these conditions, the cell acquire energy to maintain their life by cannibalizing their own organelles. Okay, so the cell will start degrading. It will start degrading its own organelles so that the metabolites can be provided for survival of the organism. Okay. Okay. You now answer this question. The nature of the lysosomal lumen is what? So just now we discussed what is the nature of the lysosomal lumen. So it is acidic, right?
Acidic.
Why is it acidic? Because it is acidic because first of all that it has VATPases. So it has ATPases. So ATPases are basically membrane proteins. These are membrane proteins which actively transport. So if you look at the structure, so I'll show the figure. Okay. So yes, the answer is option C. So you see this figure here in the bottom. So you can see here this lysosome has these membrane proteins. These membrane proteins are called VATPases. So why is it called VATPases? ATPases means what? It will hydrolyze the ATP to ADP. Okay. So the enzymes will hydrolyze ATP to ADP, and the energy that is released, that energy will be utilized to transport the protons, proton molecule which is present in the cytosol into the lumen of the lysosome. So once these proton molecules are transported into the lysosomal lumen, then what will happen? So as the H plus concentration will increase, the pH will decrease. So because of the presence of this VATPases, the lumen of the lysosome is acidic. And this acidic lumen is required for these different acid hydrolyses, which are present in the lysosome. Okay, so most lysosomal enzymes are acid hydrolyses. So all these proteases, lipases, nucleases, different types of enzymes that are there. There are several of these acid hydrolyses, and these are only active at the acidic pH. And this acidic pH is maintained by what? This acidic pH is maintained by, because of this VATPases, this acidic pH is maintained, and but not all the neutral pH characteristic of the rest of the cytoplasm. So the rest of the cytoplasm has a pH of 7.2, while the lumen of the lysosome has a pH of 5. So the requirement of lysozyme hydrolysis for acidic pH provides a double protection against uncontrolled digestion of the contents of the cytosol. Even if the lysosome membrane were to break down, the released acid hydrolyses would be inactive at the neutral pH of the cytosol. So to maintain their acidic internal pH, lysosome must actively concentrate proton ions, and this is accomplished by a proton pump. So this is a proton pump which is mentioning, and this proton pump actively pumps proton inside the lumen of the lysosome. Okay. So why is the cytosol having neutral pH? So that is that you can think of as it is having a defense mechanism, so that even if the lysosome is degraded, membrane is degraded, and the contents of the lysosome come outside the comes in the cytosol. The contents of the lysosome comes in the cytosol. Even then, what will happen? It cannot degrade the cell because this acid hydrolysis will only remain active at pH 5, while the pH of the cytosol is neutral. It is it is roughly neutral. That is why this acid hydrolysis will remain inactive in the cytosol. Okay. Okay. Okay, so answer this question. Lysosomes contribute to the immune response by. So how do lysosomes contribute to immune response? So you can see this figure here. So what it is doing. So any food particle or any foreign substance that is engulfed by the cell, by any cell, so by endocytosis, these different phagosome structures that are formed, that ultimately what happens, these food vacuole or phagosomes that are formed, they merge with this. They merge with this lysosome. So once this lysosome fuse with this phagosome or these foreign substances, then these foreign substances will get degraded because of the acid hydrolysis present in the lysosome, and they will be degraded and released from the cell. So the answer is what? It will contribute to immune response by degrading the pathogenic bacteria, isn't it? So these are very easy. These are easy questions. So in week one only, the basics of biology, you can say, was covered. So there is nothing much to understand. You already know about these. So the lysozymes, lysosomes, not lysozyme. Lysozyme is a protein. So lysosome is the organelle, and it use their hydrolytic enzymes to destroy pathogens that might enter the cell. So as we know, the different types of immune cells which are present, the white blood cells, for example. So these white blood cells, like macrophages, a type of the white blood cells are macrophages, and they're part of body's immune system. So when these macrophages engulf a foreign particle by a process known as phagocytosis, then what will happen? The section of the plasma membrane of the macrophage, it will invaginate. It will invaginate and engulf the pathogen. The invaginated section with the pathogen inside then pinches itself off from the plasma membrane and become a vesicle. This vesicle fuses with the lysosome, and the lysosome hydrolytic enzymes destroy the pathogen. Okay. So these are some of the questions which are related to lysosome. Now coming to plasma membrane, you all know plasma membrane. It is a fluid-like structure, and it is composed of lipid bilayer. So you can see in this figure that there are two layers of phospholipids. So one is this is one layer, this is the second layer, and then this lipid bilayer. They are composed of phospholipids. So they are called phospholipids because they have a head group, they have a tail group. The head is formed of phosphate, and the tail is formed of the fatty acid side chains. Okay, fatty acid chain. So the lipid, this plasma membrane, if we see that they are composed, not only they are composed of lipids, but they are also composed of protein molecules. So there are two types of protein: integral membrane protein, peripheral membrane protein. So all these things can just see the figure and understand what are the different structures present. So they have proteins, they have lipids, then they have glycolipids. Okay, there are different types of proteins. So transmembrane proteins means what it will have a particular domain which remains outside the lipid bilayer, and a particular section which remains inside the lipid bilayer. Then there are peripheral membrane, means only a part of the protein, or it will be present only on one side of the lipid bilayer, either on the outside or it will be present on the inside of the lipid bilayer. So yeah, the plasma membrane consists of both lipids and proteins, and the fundamental structure is the lipid bilayer. And the proteins are embedded within this lipid bilayer, which carry out specific functions. So what is the function of the proteins? So the function of the proteins which are embedded in the lipid bilayer is selective transport of molecules and cell recognition. So there are different types of proteins, right? So there are channel proteins. Then there are um, pumps. So the pumps, just now we saw one of the pumps in lysosome. So the pumps will actively transport a molecule from one side to the other side. Then the channels freely allow free passage of molecules from one side of the one side to the other side of the membrane. Okay. So then there are and another function is cell recognition. So there are different surface markers that allow one cell to recognize the other cell. Okay, so the membranes of animal cell contain 5 major phospholipids. These are phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylainositol, sphingomyelin. So these are different types of phospholipids, and the lipids account for 50% component of the plasma membrane. And these phospholipids are asymmetrically distributed between the two halves of the membrane. Why are they asymmetrically distributed? Because you can see here that the composition of these different phospholipids. Phosphatidylserine, phosphatidylethanolamine, phosphatidylainositol, they are not uniform on the two uh, on the in the two leaflets, in the outer leaflet and the inner leaflet, they are different. So the outer leaflet may have a one type of phospholipids in more percentage, while the inner leaflet may have a particular phospholipid component more. Okay, so that is why it is called asymmetric distribution. Okay, the outer leaflet of the plasma membrane mainly consists of this phosphatidyl and sphingomyelin. So just I was mentioning this only. So phosphatidyl, ethanolamine, and phosphatidylserine, they are mainly present in the inner leaflet. While the outer membrane mainly has phosphatidylcholine and sphingomyelin. Okay. So these are some few of the points, and the plasma membrane of animal cells also contain these glycolipids and cholesterol. Okay, so to answer this question, just now we discussed the plasma membrane contributes to what? What will be the correct option? So how the plasma membrane contributes to cellular recognition? By synthesizing proteins, by transporting ions through recognition proteins that identify other cells by producing ATP. So the fluidity of the plasma membrane is provided by what? It is provided by lipid molecules, while the recognition is provided by what? It is provided by the synthesizing proteins, right? Just now we mentioned we were discussing the different points, important points of plasma membrane. That these proteins, surface proteins, surface markers, biomarkers. We also call them as biomarkers, like particular protein that is present in that is overexpressed in a particular cell, but it is not present in the other cell. So that helps in cellular recognition. So the proteins are responsible for carrying out specific membrane functions, and most plasma membrane consist of approximately 50% lipid, 50% protein by weight, with the carbohydrate proportions of glycolipid and glycoprotein constituting 5 to 10% of the membrane mass. So since proteins are much larger than lipids, the percentage corresponds to about one protein molecule for every 50 to 100. So these are some extra information. I just put them for your information. So around 50% lipid and 50% protein by weight is present. Okay. But the proteins are much larger. That is why. So by weight, if we see, then the amount of lipid and amount of protein is 50-50. But the percentage of protein is one every 50 to 100 molecules. Okay. So there are different functions of plasma membrane where there are transport. They can act as transport proteins like the sodium potassium transporter. They can act as receptors. So these receptors are also helping in cell recognition, like LDL receptor. And then there are enzymes So the different proteins that are present on plasma membrane, they can be transporter proteins, they can be receptor proteins, they can be enzyme proteins, and there can be signal transducer like insulin receptors, uh, G protein coupled receptors. All these are different signal transducers. So for helping transferring the information in the environment to the inside of the cell. And there are different functions like support, protection, communication, all these things okay. So now answer this question, which of the following is true with respect to the fluidity of the plasma membrane? So on so these are the options. So there are several statements which are present in this question. So as temperature increases, it increases fluidity and increase transport of protein and ions across membrane. As temperature decreases, it increases fluidity and decreases transport of proteins and ions across membrane. As the saturation of fatty acid increases, it decreases the fluidity of the membrane. As saturation of fatty acid increases, it increases the fluidity of the membrane. So which of the above statements are true? So these concepts, fluidity of plasma membrane and things, you must have studied in cell biology, biochemistry courses. So what determines the fluidity of the plasma membrane? So just now I was discussing that the lipid molecule is composed of a phosphate head group. So it has a phosphate head group and it has a fatty acid chain. So the length of the fatty acid chain and the degree of saturation. So what is the length of this fatty acid chain? And what is the degree of saturation? Degree of saturation means what? That how many double bonds or multiple bonds are present in the fatty acid structure. Different types of there the structure of a fatty acid. There are different types of fatty acids, oleic acid, linoleic acid. These are some of the unsaturated fatty acids. So unsaturated fatty acids means what? It will have double bond. So if the more the number degree of saturation or the more the number of double bonds, the more fluidic it will make the membrane more fluidic, and the larger the length of the fatty acid chain, the more compact it will be. Okay. So as the saturation of fatty acid increases, it decreases the fluidity of the membrane. So saturation increases means what? So saturation means the number of single bonds. Okay. As the saturation increases means the number of single bonds are increasing. So if the number of single bonds are more, then it will obviously decrease the fluidity. If the number of double bonds are more, it will increase the fluidity. Okay. So degree of saturation means what are the number of single bonds? So like CH3, CH2, CH2, CH3. So it has three single bonds in this parent chain. So if there are double bond CH, CH3, so it has a single double bond. So this will, if such structure is present, it will make the fatty acid more fluidic. Okay, because it introduces a kink in the structure. Okay, so answer is actually option A, 1 and 3. As the saturation, this option is wrong because as the saturation will increase, it will decrease the fluidity. So this is correct option. And as the temperature increases, obviously what will happen? It will increase the fluidity and increase transport. So option 1 and 3 are correct. So since statement 1 and 3 are correct, option A is correct. Okay. So these are some of the things which we can skip for now also because these are not really required in this course. So there are vesicular transport means how this molecule which is synthesized outside the cell is entering or reaching different specific organelles inside the cell, or how a particular protein which is synthesized in the nucleus or which is synthesized in the ribosome which is present in the cytoplasm, how does it reach different parts of the cell. So what are the different organelles? Like endoplasmic reticulum, Golgi complex, endosome, lysosome, vacuole. So these are the different organelles which are involved. So together they form an endomembrane system. So endoplasmic reticulum transfers their protein to a Golgi, and then the Golgi several protein modifications will take place. Then this will be transported to another organelle, endosome, then to lysosome, vacuole, and this way his vesicular transport takes place. These are in quite amount of detail in cell biology. Any classical cell biology textbook will not go into too much detail. Just a little bit brief about the Golgi complex. So the Golgi complex is present. So outer to the endoplasmic reticulum, you can consider that this Golgi complex is present, and this Golgi complex, similar to the endoplasmic reticulum, also it is made up of several cisternae and tubules. So these are the cisternae and these are the tubule structures. And so there is again a division of this Golgi complex. So the cisternae or the tubules which face the endoplasmic reticulum, they are called the cis facing. Then there are median Golgi complex. Then the trans facing are the Golgi complex, which is facing the periphery of the plasma membrane. Okay, so the proteins that are received from the ER, they are processed and sorted for transport to their eventual destination, lysosome, the plasma membrane, or secretion. So the RER, basically the rough endoplasmic reticulum, is synthesizing different proteins. Now these proteins have to be transported to different organelles. So for example, your mitochondria requires a particular protein. For example, porine protein. Just now we were discussing about the porine protein. So for example, a porine protein is synthesized by ER. Now this porine protein has to be transported from the ER to the mitochondria, or say a acid hydrolase. Acid hydrolase that has been synthesized inside the endoplasmic reticulum, that has to reach the lysosome. So how are these transport taking place inside the cell? Okay, so what happens first? This protein that has been synthesized by ER, that reaches the Golgi complex. It is, and in the Golgi complex is also called the post office of the cell. So in a town, a post office, what it does, it segregates the different different letters or postcards that is received. That, for example, a particular postcard has to reach north, has to reach south or east or west. So they're segregated. So similar function is done by this Golgi complex. Okay. So they are processed and sorted for transport to their destination. So a particular protein might be needed in the lysosome, particular protein might be needed on the plasma membrane, or there can be proteins that needs to be secreted out. So these sorting is done by this Golgi complex, and this Golgi complex is composed of flattened membrane enclosed sacs, which are called cisternae, and this cisternae is associated with vesicles. So the proteins which are facing the endoplasmic reticulum, they are called the cis phase, and it is oriented towards the nucleus. They are then transported through the Golgi and exit from its trans phase. So from the trans phase, it is exited, and the proteins are carried through the Golgi in the cis to the trans direction. So the proteins will always be carried from the cis to will move to the medial. So these vesicles also, these vesicles are actually burned off. So a part of the membrane of the Golgi complex burned off. They form these vesicles, and then they go and fuse to the other part of the Golgi complex, to the median Golgi complex, and then in this way the transport takes place. Okay. Then several modification also takes place, like mannose 6-phosphate modification, and there are several carbohydrate groups that are attached to protein. So all these, there are different, there are much detail. We can go into any depth. So let's not discuss so much. So the different modification, just remember the function, the different modification of protein, mainly the O-link glycosylation. In the endoplasmic reticulum, N-link glycosylation takes place. This is O-link glycosylation takes place, and the sorting takes place in the trans-Golgi network. Okay. So these are some of the functions. So what are these are again a few statements. You need to read these statements carefully and then answer. So what are the functions of endoplasmic reticulum? And you have to read the question carefully, actually, because the question is which of the above are false. Okay. So the first statement is what? They are site for protein synthesis for secreted and integral membrane proteins. So this is the correct statement because the endoplasmic reticulum is a site of protein synthesis. They are site of bulk membrane lipid biogenesis. This is also true because SERC, smooth endoplasmic reticulum, is involved in membrane lipid biogenesis. Now, protein and phospholipids are transferred and biochemically modified in the region of the endoplasmic reticulum that is in close to the Golgi apparatus. So biochemical modification of proteins do take place in the endoplasmic reticulum, like protein folding and uh, several modifications take place in the ER as well. They are major storehouse of intracellular calcium. That is also true. And the endoplasmic reticulum is composed of continuous membrane system that includes the nuclear envelope and peripheral ER. This is also true because ER, if you see that the outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. So it includes both the nuclear envelope and the peripheral. And the modifying, sorting, and packaging. So modifying, sorting, and packaging of proteins for secretion is a function of Golgi complex. Okay. So option numbers, the statement C6, statement number 6 is false. So the answer is option D. Okay, so you need to understand the function of different organelles. So a little bit understanding is necessary. With the primary function of Golgi apparatus in vesicular trafficking is what? So synthesizing protein is a function of ER. So this is wrong. Degrading cellular waste is a function of lysosome. This is false. Storing cellular energy is a function. Mitochondria. So option C is false. The only option which is correct is modifying and packaging proteins. Okay. Okay. Now the last section of today's session, what is differential centrifugation and density gradient centrifugation? So can someone, if anyone has gone through the lecture, can you explain that when is differential centrifugation required and when is density gradient centrifugation required? What is the difference between the two? So there are two types of, if you want the components of the, want to fractionate the components of the cell, then there are two techniques by which you can fractionate it. One is differential and another is density gradient. So which is what? And when is one required over the other? So basically, differential centrifugation is what? So it is if you want to separate the different components of the cell based on their sedimentation rate. So okay, so different components of the cell, like all the organelles that we studied just now, nucleus, the mitochondria, ribosomes, lysosome, endoplasmic reticulum. So all these have a particular density. Okay, they have a particular density, they have a particular sedimentation rate, or size of one is larger compared to the other size. So based on their size and based on their sedimentation rate, if you want to separate these different organelles. So for example, for a particular experiment, you want to isolate the protein which is present. So you want to work on nuclear lamines. You want to understand a particular protein which is present on nuclear lamine and how it what is the functional role which is played by that protein. So what you have to do? So first of all, you have to isolate the nucleus from the cell. So what will you do? So for that, you can go for differential centrifugation. So what is differential centrifugation? So basically, you are separating the components of the cell on the basis of their size and their density. Okay, so first of all, what you have to do is you have to lyse the cell. You have to lyse the cell means you have to rupture the plasma membrane of the cell. So this is called homogenized cells. So there can be different. There are different mechanisms which are followed in lab, like physical mechanical disruption, like you use a sonicator, or in physical methods you use heat shock cycles, like freeze and thaw cycles. You cool it and then you heat it. And in this way, what happens? The plasma membrane gets ruptured. Or there can be chemical methods, like you use for degrading a bacterial cell wall, you can use uh, an enzyme that acts on the peptidoglycan. Okay, or chitinase for disrupting. So these are chemical. Enzymatic means the same. Uh, okay, so enzymatic means you are using enzymes. Chemicals like any chemical that can rupture the bacterial membrane or a human cell membrane. Okay. Then what is done? The cells are centrifuged. So ultra centrifuge means you have to go above 100,000 RPM. So look at this figure carefully. So first you have created a you have homogenized the cell. Okay, so once you have homogenized the cell, you have a cell suspension. Okay, now in this cell suspension, there are different cellular components. Now, what happens if you centrifuge this cellular suspension at 800 G, 800 G. So there are two different units of. So if you have used a centrifuge in your laboratory, then you must be knowing that there are two different units. One, you can either explain it in the form of RPM, or you can define it in terms of G. 800 G means 800 times the force of gravity. Okay, so you are centrifuging it at 800 G for 10 minutes. So what will happen? So at this centrifugal force. At this centrifugal force, the nuclei being more heavier or having greater size, they will settle first. They will form a sediment. Okay, they will form a pellet. So 800 G in the sediment, what do you get? You get nuclei, and in the supernatant, you have the other components. Now, what you do, you collect out this supernatant. Separate this supernatant, and then you subject it to another round of centrifuge. So you separate this supernatant, and then you centrifuge it again for 15,000 G for 10 minutes. So once you do that, then you again get a pellet. Now, in the pellet, what do you get? In the pellet, you will have this mitochondria, lysosome, peroxisomes, all these will be present. Okay, now again you will get a supernatant. You take out this supernatant again, and then you subject it to a higher centrifugal force, like 100,000 G for 60 minutes. You are increasing the centrifugal force at each step, and you are increasing the time as well. So why you are doing that? Because these are having lesser, they are having lesser density. They are having lesser size, less size. So once you do that. So once you centrifuge it at 100,000 G, then the even the smaller dense or the smaller size particles like these that you see here, the endoplasmic reticulum sediment, all these things start sedimenting, and then you take out the supernatant, and again you spin it for at a higher centrifugal force at 200,000 G, and in the end that you get the ribosomes. So the ribosomes are having the least density. So you need greater centrifugal force for isolating the ribosomes. Okay, so it is good if you remember the order. So at lower RPM, you will first get nucleus. Then what do you get? Once you increase the centrifugal force, you will get the mitochondria, lysosomes, peroxisomes. Okay, mitochondria, lysosome, peroxisome. Then in the second round of centrifugation, what do you get? You get this endoplasmic reticulum sediment, and in the fourth step, you get the ribosome. So this is differential centrifugation. So this is a rough centrifugation. So you will not get very pure sample, but you can isolate the different components to some extent. So roughly you can separate the different components. So that is differential centrifugation. But if you do density gradient centrifugation, so like in this step that you see, that you can see here that the mitochondria, lysosome, peroxisomes, all these components are present together. So they are having roughly the same size. That is why you are getting them together. Okay. But if you do a density gradient centrifugation, then you can further separate these components, okay, based on the uh, uh, particular density. So once I discuss what is density gradient, then you will understand this. Okay. Okay, so this question I forgot to add the animation again. Okay, so the following organelles that will let down at 15,000 G for 15 minutes are. So now these values and figures, they are actually difficult to remember, but you need to have a rough understanding. So remember, you see, if you remember this figure, that at 800 G, the pellet that you were obtaining was that of, okay. Now, when you are spinning at 15,000 G, the pellet that you obtain is that of these mitochondrial lysosome and peroxisome, isn't it? Mitochondrial lysosome, peroxisome. So the answer is option B. Okay. So when you spin it at 800 G, then the pellet that you get is of the nuclei. And when you spin it at 15,000 G, the pellet that you get is that of these mitochondrial lysosome, peroxisome. And then what is the higher, even higher centrifugal force that is required? 100,000 G, means 1 lakh G. Okay. So at 100,000 G, what do you get then? You get this ER, like microsomes and all these things. So the ER basically gets fragmented to form microsomes, and you are left with this fragments of plasma membrane and endoplasmic reticulum. Then after 100 G, you go for 200 G. So at 200 G, what do you get? 200,000 G, you are left with ribosomes. Okay. Okay. So this is one of the figures from the lecture slides. Okay, and the figure that I discussed, that is there in your this classical textbook, Cooper. So it has been very nicely explained in that textbook. So you can go through those as well. Now, here also there is another question related to differential centrifugation. So in differential centrifugation, so in a certain study performed by a group of students on an unknown cell type, yielded the following fractions. Okay, so you have a particular cell type and you are getting the following fractions. So A, B, C, and D on 4 steps of centrifugation. So the fraction A is having the, so there are 4 steps of centrifugation at 600 G. So the first is at 600 G. Then you are doing at 100,000 G, or 1 into 10 to the 4 G, actually 10,000 G, not 100,000. This is 10,000 G, then 100,000 G, and then 32,000 G. So now the fraction A, which is having the highest mass. So A is having the highest mass. So which of the organelle has the highest mass? Which organelle has the highest mass? This is having the highest mass. Okay. And the B is having the lowest mass. So just now I explained. So now answer this question quickly. This is having the lowest mass. So what you have been asked in this question is the fractions C and D have intermediary masses with C greater than D. Okay, C greater than D. So A and B, then C, D. So this is the order of the size of the mass of this fractions. Okay, mass or size. So now we have to say, you have to answer that what is A, what is B, what is C, what is D. Okay, so A is having the highest mass, which means A is what? Anyone? So did you all understand the concept of differential centrifugation? Is what? So the one which is having the highest mass will form the sediment fastest. Okay, so the highest mass is what? Which organelle has the highest mass? It is the nucleus. And which organelle has the lowest mass? It is the ribosome. So ribosomes have the lowest mass. That is why you need to go till 32,000 G, or 3320. So this is 3,20,000, not 32,000. This is 3,20,000, or more than 200,000 G for getting for separating the ribosomes. Okay, so you need a ultracentrifuge for carrying these experiments. Okay, so which is the correct option now? So now you can see option C is the correct because A is nucleus and D is ribosomes. Rest you can ignore. So just remember which is the organelle which is having highest mass. It is nucleus, and the organelle which is having lowest mass is the ribosome. That is why just the highest and lowest, if you know the information, then you can solve this question. Okay. Option is B. Now, what is the use of detergents in the process of differential centrifugation of cells? So why do you use detergents? Basically, detergents are required for membrane proteins. Okay, because if you look at the structure of detergents and if you look at the structure of the plasma membrane, then the detergents also have a polar head and a hydrophobic tail. So the stability, the membrane proteins are provided stability by the structure of the detergents. Okay. Okay. So the answer is actually to lyse the plasma membrane and extract cellular organelles. So that will be the correct answer, isn't it? Answer is option 3 or option C. Okay. So that is all for today. So I hope all these points were clear. So are there any doubts related to today's session? Any particular section which you didn't understand? Or is it clear? Okay, so this session had, it had very less. So Nanti Vasoya, do you have any question?
Yes, sir. I
Yeah.
just enrolled in the course today. So where can I find the recording videos of the previous lectures?
Recording videos of the so you want recording of this session or the that has been
Last
taken
week.
by the course last week. So you can directly log into your Swayam NPTEL portal, and then you can there you will this week one lectures have already been uploaded, so you can find the lectures there, or
Okay.
or uh, or you can directly type on YouTube also. In YouTube also these lectures are there. So NPTEL Genetic Engineering, if you directly search on YouTube, then also you will find the lectures are there.
Okay.
But
Thank you, sir.
yeah, yeah. Okay. Okay, so anything else? Okay, so if there are no more questions, then we will again meet in the next session next week. So till then, go through your week 2 lectures and solve the assignments. If you have any doubt in any of the assignments, or try to solve the week 1 assignments. And then if there are any doubts, then we can take up those questions as well. Okay. Okay. Thank you all for patiently listening. So I will end the session now.